Hi class, in this recording we're going to focus on homeostasis. When we think of homeostasis, it's the idea that we have the internal environment of our body is relatively unchanging. It's going to be relatively constant. And a key phrase here is relatively. We can have minor fluctuations, minor increases and decreases to whatever variable it is that we're trying to maintain. For instance, our body temperature can go up or down a little bit. Our blood sugar content can go up and down just a little bit, but they need to hover around a predefined set point. And when we think of feedback or homeostasis, negative feedback is the most common kind of homeostasis. It allows for this dynamic equilibrium, this hovering of a variable around a predetermined set point. And one of the key distinctions of negative feedback is our body's response is always going to be opposite to the stimulus. So this for instance, if our body temperature is increasing, so we have if our body temperature is going up, our response will be to lower our body temperature by sweating. We're always going to try to return to the set point with our negative feedback to maintain homeostasis. If we have a loss of homeostasis, we could have a disease set in or disease state, and if the loss is profound, we could have death occur. So let's talk about a blood pressure as an example of negative feedback. So we have receptors, and the receptor is going to measure the variable. It's any structure in our body that measures the internal environment of our body. We have many different kinds of receptors. Receptors for pressure, receptors for pH, receptors for sugar levels, so on and so forth. And then we have an integration center that processes the information and makes a decision, and then sends that decision to the effector. The effector is going to initiate the response based on the decision made by the integration center. So when we think of our integration center, almost always this is going to be our brain. Occasionally it's going to be our spinal cord. And the effector is almost always going to be some sort of muscle or glandular structure in our body. So let's look at blood pressure as an example. So somebody stands up, they're snoozing in bed and they stand up quickly. And because they stand up quickly, blood drains out of their head and they lose homeostasis. The baroreceptors in their heart, and the baroreceptors are a kind of sensory receptor that measures blood pressure, is going to record the drop in blood pressure and then send that signal to the integration center. In particular, the brain stem of our body will receive that signal. And in response to receiving a drop in blood pressure, the brain stem will send a signal to the heart that accelerates our heart rate. And as the heart starts to pump faster, we're going to send more blood to the brain, and that will cause blood pressure to rise at our brain and restore homeostasis. We also could have positive feedback. And when we think of positive feedback loops, they tend to be a little bit more exciting for most people. They also tend to be a little bit scary for me because of the amplifying effect. So when we think of a positive feedback, I want you to think of a rapid change that can occur within our body. And these positive feedback loops are going to amplify a response. When I talked about negative feedback, the response was always going to be opposite of the stimulus. With a positive feedback loop, the response is going to be the same as the stimulus. So our response is the same as this instead of the opposite. And this causes the stimulus to get stronger and stronger and stronger, which causes the response to become stronger and stronger and stronger. Classic examples of positive feedback loops include childbirth, blood clotting, protein digestion, and action potentials, or sending electricity down a nerve fiber. These positive feedback loops can be dangerous, and they can be dangerous because they can amplify a signal out of control. For example, we could have a runaway fever, and you have to break the cycle in order to break the positive feedback loop, and that typically requires a very dramatic change. So for instance, let's look at childbirth. So here's a baby in utero getting ready to be born, and that baby presses his or her head up against the uterus. And then as the baby does that, that's causing the cervix to stretch, the opening of the uterus to stretch out. That stretching force will send an impulse to the brain, and then the brain is going to be stimulated to release oxytocin. Oxytocin will then travel to the uterus and cause the smooth muscles of the uterus to contract more. That causes the head to push on the cervix even more. That causes more stretch signals to be sent to the brain, which causes even more oxytocin to be released, which causes even more pushing of the child against the cervix, and it just keeps going around and around and around as a positive feedback loop. To break the cycle, the child needs to be born. And if that child is born, then the stretching on the cervix stops. In our bodies, we also are going to have gradients. Gradients, particularly concentration gradients, are important for dictating the direction that chemicals will diffuse. Pressure gradients are important for dictating the direction that fluids will flow. So as we look at these gradients, we can have chemical gradients based on chemical concentrations and pressure gradients. Occasionally we have the temperature gradients that we talk about in this class, but we don't spend a lot of time talking about temperature gradients. One of the laws of thermodynamics is that substances have a natural tendency to spontaneously go from an area of high concentration to low concentration, or from high pressure to low pressure. So think of a kid riding a wagon down a hill. Kids riding a wagon down a hill, generally speaking, will flow downhill or move downhill from an area of high potential energy to an area of low potential energy. But to go back up the hill requires an input of work. That's not a spontaneous process. When we move with the gradient or down the gradient, that's spontaneous and does not require the input of any energy. However, to move against the gradient, or in this instance, to move up the hill, that requires the input of energy. It's not a spontaneous process. And in our bodies, we will expend a lot of metabolic energy moving chemicals and fluids against their concentration gradients. So as we look at our chemicals, for example, we could have glucose up here in part A of this figure. Glucose has a high concentration outside of the cell, a low concentration inside of the cell, and will spontaneously want to move or diffuse into the cell. As we look at charged particles are typically going to flow with their electrical gradient. So if we have positive ions on the outside of the cell and mostly negative ions on the inside of the cell, those positive ions will be drawn in with the electric charge. As we think of heat, heat will flow from areas that are warmer to areas that are colder. So we have a tendency, since we're warm-blooded animals, to release body heat to the cooler environment. When the temperature outside is 98 degrees or body temperature, we have a hard time releasing heat, and we start to overheat very rapidly in those temperature environments. That's all we have for this recording. If you have any questions or comments about homeostasis or gradient movement, please feel free to post them on the class discussion boards or to shoot me an email. As always, happy studies.